Warm greetings to one and all watching this video. I'm here to present my research work entitled as Formulation Development and In-vitro Characterization of Cyclosporine-loaded ocular inserts for dry eye disease. So dry eye disease is a multifactorial condition that occurs due to any dysfunction in the glands that are responsible for the formation of the tear film. The tear film is basically composed of three layers, namely the lipid layer, the aqueous layer, and the mucin layer, and any dysfunction in one of the three layers would result in an increase in osmolarity of the tear film. This eventually leads to a cascade of immune responses such as epithelial cell membrane disruption, damage to the goblet cells, alteration in the protein-lipid interactions of the tear film, and triggers a release of inflammatory cytokines, causing ocular irritation and inflammation. Cyclosporine is well known for its anti-inflammatory effects and it has been intended for the management of dry eye disease. However, the currently available marketed formulations are in the form of nanomicellar solutions and emulsions, and these are associated with numerous limitations such as low ocular viability and a high frequency of administration. Here, we intend to develop inserts to reduce the frequency of administration and enhance patient compliance. The drug and the polymer are fed into a hot melt extruder, and the resulting extrudes are then fed into a stretching and cutting machine called the calicut that gives us the inserts of desired dimensions. Optimization of process parameters of both HME and the stretching and cutting machine were carried out. Initially, we studied the effect of pre-mixing step on the content uniformity. As we all are aware that a large quantity of drug usually goes into HME, as in the case of amorphous solid dispersions, where around 15 to 40% of the drug is loaded in a polymeric matrix. However, for ophthalmic indications, the dose required is generally low, and here they have incorporated only 1% of the drug. So achieving content uniformity was a major challenge. We compared three different pre-mixing steps, namely geometric mixing, sieving, and wet mixing, and we found that wet mixing yielded inserts of good content uniformity. Then we also optimized the barrel temperature and the screw speed. Here we used the one factor at a time approach where initially the screw speed was kept constant and the temperature was altered to identify the optimum temperature for good extrudability of the inserts. Then we kept the temperature constant and the screw speed was altered. So the table here depicts the optimum temperature for good these molecular weights of the polymer hydroxypropyl cellulose. We also studied the effect of the screw speed on in vitro release, and we found that there was no significant difference in the release profile. So this in turn establishes the robustness of the mixing that occurs inside the barrel of HME. Here we also study the effect of the heater temperature of the stretching and cutting machine on the sharpness of edges. So basically, though HPC is approved by the FDA for ocular indications and it is biocompatible, here we can define biocompatibility not based on the material property but based on the surface characteristics of the dosage form. So here we could see that when the operation was carried out at 25 degrees Celsius, the edges of the inserts were sharp. However, when the same unit operation was carried out at higher temperatures, say 45 and 55 degrees Celsius, inserts of relatively smooth edges were obtained, which would be more compatible than the previous ones. Then, after fabrication of the inserts, evaluation was carried out. The weight of the inserts were around 5 mg and the average dimensions were 3.3 into 1.27 mm. The surface pH of the developed inserts were 7.3, that was in accordance with the ocular pH. The drug excipient compatibility was carried out by ATR spectroscopy, and here we could see that the characteristic peaks of both cyclosporine as well as HPC is retained in the ATR spectra of the formulation. So this indicates that there were no significant interactions between the drug as well as the polymer, and that is why the peaks have been retained in the ATR spectra. The inserts were also characterized for swelling index because swelling is an adhesiveness of the inserts once it is placed inside the cul-de-sac. The results of scanning electron microscopy also revealed the smooth surface of the inserts. So, since there is no compendial method available for the dissolution testing of ocular inserts, we developed a novel in-house in vitro dissolution method. Here, we placed a stainless steel mesh in between the magnetic bead and the insert in order to prevent accelerated dissolution of the inserts. In the absence of a stainless steel mesh, what we observed was that the magnetic bead would often interfere with the dissolution profile of the inserts, giving us a false profile. However, when we developed the mesh with certain perforations and spaces, we were able to find that efficient circulation of the media was obtained, and this method was also able to discriminate the dissolution profile of various molecular weights of the polymer, hence enabling us to achieve the identification of product to product variability. So for the selection of dissolution media, initially we carried out equilibrium solubility studies, as we know that there is a criteria that the dissolution media should be able to dissolve 3 times higher the quantity of drug that is present in the formulation in order to maintain sink conditions. We selected 0.25% SLS as our final media as it exhibited the highest solubility. Based on the release profile that was obtained in both 0.1% SLS in PBS versus 0.25% SLS, we could see that 0.25% SLS gave a better release profile, and therefore we have selected that dissolution media. Further, the volume of the dissolution media also is critical here because only 50 microgram of the drug was loaded into the formulation, and using a higher volume of the dissolution media would be challenging for detection through HPLC, and hence we restricted to only 5 ml of PBF. This is the in vitro release profile of the inserts, and here we could see that the MXF grade of HPC, that is around 850,000 Daltons, was able to give a sustained release of the drug for a period of 24 hours. And also the developed dissolution method was able to differentiate between the release profiles of various other molecular weights, thus proving the discriminatory power of the dissolution method that has been developed. Here, the in vitro degradation of the inserts are also in accordance with the release results. Here we could see that the MXF grade was degraded at 24 hours, where only less than 10% of the polymer was remaining. So this is in accordance with the in vitro release profile that the MXF grade is ideal for fabricating inserts that are intended for once in a day administration. The inserts were also characterized for stability over a period of 3 months, and here we could see that there was no significant change in the percent drug content after 3 months of storage at both room temperature as well as accelerated conditions. The not significantly different. They were established. Vivo eye irritation studies were carried out where one eye of the rabbit was left untreated that served as a control, and the other eye was the treatment. And based on race scoring system, we could see that there were no significant differences in both control and treatment, and hence score. So I like to conclude by saying that the developed cyclosporine-loaded ocular inserts were able to give a sustained release of over 24 hours and were found to be stable at 3 months of storage. And the in vivo biocompatibility studies also exhibited no eye irritation. So the developed formulation could be used as an alternative to marketed formulations, as it has a low frequency of administration, that is just once in a day, as compared to the marketed ones which needs to be administered 4 to 5 times a day for management of dry eye disease. Thank you.